Hello, class. We're doing our lecture on chapter 16, the lymphatic system and immunity. Two concepts very closely together, so that's why we're going to cover it in the same lecture, same chapter. So let's just get started. Okay, so today we're introducing the lymphatic system. This is a body-wide network of vessels, tissues, and immune cells that travel alongside the cardiovascular system. Functionally, it does three big jobs. First, it returns excess interstitial fluid. So that is the fluid that leaks out of blood capillaries back to the bloodstream, which prevents tissue swelling and it helps maintain normal blood volume. Secondly, number two, it absorbs dietary lipids from the small intestine and transports those fats into circulation. And third, number three on the big list, it defends the body against disease. So the lymphatic system houses many of the cells that recognize, attack, and remember pathogens. So you will also hear the term immune system. That's because a large portion of immunity is carried out by lymphatic cells, notably lymphocytes, and phagocytes that reside in our circulate through lymphatic structures. So, in other words, the immune system isn't a single organ, it's a functional system embedded in lymphatic tissue and organs distributed throughout the body. So by the end of the chapter, we'll be able to trace lymph from tissues back to the blood, identify where immune cells are produced and activated, and explain how the structure supports function through lymphatic pathways. Okay, lymphatic pathways. So let's map the route lymph takes through the tissue back to the veins. Fluid first enters the lymphatic capillaries. Now, these are microscopic, close-ended tubes made of simple squamous epithelium that lie parallel to blood capillaries throughout the body. Once interstitial fluid enters these capillaries, it is now called lymph. From here, lymph flows into progressively larger lymphatic vessels. And these resemble veins but have thinner walls and valves to keep lymph moving in one direction. These vessels deliver lymph to lymph nodes where it is filtered and surveyed. Leaving the nodes, lymph passes into larger lymphatic vessels still that converge into regional lymphatic trunks. Now, these are named for the territories they drain. We have the lumbar trunk, the intestinal trunk, the intercostal trunk, the bronchomediastinal trunk, the subclavian trunk, and the jugular trunk. The trunks empty into one of two large collecting ducts. The thoracic duct, which is the longer and wider of the two, it carries more of the lymph and drains most of the body and empties into the left subclavian vein. Then we have the right lymphatic duct, which is smaller and drains the upper right portion of the body into the right subclavian vein. Finally, lymph rejoins the bloodstream at these venous angles. This entire pathway ensures that fluid homeostasis is restored. We don't build up fluid in our body that is leaked. And the pathogens collected from tissues meet immune checkpoints along the way. Okay, so here's a picture that is a zoomed in picture of the lymphatic capillary. So what we have here is we have the venu and the arteriole, which is very similar to the microscopic picture of capillaries we saw in last chapter when we saw the green lines underneath. Well, now we're seeing what those green lines are. So as we move into the capillary beds where fluid is exchanged, we can see how these lymph vessels are intertwined with the blood capillaries. And so, they're going to mesh and they're going to bob and weave and they're going to pick up any fluid that has been leaked in the exchange of the blood capillaries. So after lymph enters the capillaries, it moves to lymphatic vessels, which look like veins but have thinner walls and a pronounced system of semilunar valves. Histologically, their walls have three layers. They have the uh, endothelial lining, which is a middle layer and with smooth muscle and elastic fibers, and an outer connective tissue layer. The valves are crucial because lymph is low pressure. It's picked up at the capillary sites. They enforce one-way flow. This prevents retrograde movement as lymph ascends towards trunks and ducts. So smooth muscle in these vessels can gently contract to help propel lymph, especially in the larger vessels, while external forces such as skeletal muscle contraction and respiratory pressure changes provide the primary pumping action. Larger lymphatic vessels lead to lymph nodes. And this is where lymph is filtered and pathogens are exposed to lymphocytes and macrophages. From there, lymph continues into an even larger collector and route to regional trunks. Okay, we talked about those trunks already, and we will continue to talk about these trunks in a little bit in the future slides here. Okay, here we go. Regional lymphatic trunks, they collect the lymph from large body territories. Their name reflects their geography. We have lumbar, intestinal, intercostal, bronchomediastinal, subclavian, and jugular. So these trunks converge into two collecting ducts. The thoracic duct, which is the longer and wider channel that drains the majority of the body, including both lower limbs, the abdomen, the left thorax, the left upper limb, and the left side of the head and neck empty into the left subclavian vein. The right lymphatic duct is much smaller and drains the upper right quadrant. So that's the right side of the head and neck, the right upper limb, and the right thorax, and it drains it into the right subclavian vein. This asymmetry is a classic test favorite, right? So definitely kind of know this portion because it might show up again. All right, so here's just a picture of what we were just talking about. So the right lymphatic duct covers the right upper side of the body and the limb, and then everything else is going through the left subclavian vein. And on the picture B, we can see how these uh, lymph nodes and lymph vessels, they attach to this vein where they'll drain in. Okay, so just a recap of the pathway. So tissue, fluid, and lymph. Let's talk about the actual fluid. So lymph is simply tissue fluid that has entered a lymphatic capillary. Its formation mirrors capillary dynamics. So blood pressure filters water in small solutes. Plasma at the arterial end of blood capillaries generating the tissue fluid. This fluid closely resembles plasma, but lacks most plasma membranes, as plasma proteins. Sorry, not plasma membranes, plasma proteins. And it lacks these because these proteins are just simply too large to filter freely through these vessels. So when tissue fluid enters the lymphatic capillary, it becomes lymph, and then it begins its return journey. So what's actually in lymph? Okay, it's water, dissolved nutrients, gases, hormones, tiny protein that need to be reclaimed, and whatever foreign material is happens to be there. It could be cell debris, could be microbes, that kind of stuff. The system depends on a balance. It depends on this balance. If filtration exceeds lymphatic uptake, edema ensures. So you might get a collection of fluid around your ankles. If lymphatic are overactive or inflamed, local immune responses can be heightened. So functionally, this conversion from tissue fluid to lymph ensures volume homeostasis, protein recycling, and immune health. So as fluid moves along, the lymph nodes act as filters and immune checkpoints where lymphocytes can activate and the macrophages can phagocytose debris. All right, so that is tissue fluid and lymph. So now let's talk about how this lymph flows. So lymph flows under very low hydrostatic pressure. So much like venous blood, it relies on several auxiliary pumps. First, skeletal muscle contractions rhythmically compress lymphatic vessels, and this helps to propel the lymph forward. Valves prevent backflow and segment the column for efficient movement. Secondly, respiration creates a pressure gradient. When you inhale, thoracic pressure drops while abdominal pressure rises, pulling the lymph upward from the abdomen into the thorax. Basically, think of every breath as a gentle lymphatic suction. So third, the smooth muscle in large lymphatic vessels can contract, adding a slow intrinsic peristalsis. Because these mechanisms are actively dependent, lymph flow is highest during exercise and can stagnate with immobility. Clinically, this explains why movement, deep breathing, and compression strategies support lymph return in patients with lymphedema or other lymph node dissection. The system's valvular architecture is also protective. Even when external forces reverse momentarily, valves lock in forward to progress. So in sum here, movement is really a team effort. The muscles, breathing, vessel tones, and valves, they all are coordinated to move a delicate low pressure fluid uphill to the venous circulation. Okay, the function of lymph. So functionally, lymphatics do four major things in transit. Number one, they absorb dietary fats from the small intestine and deliver them to the bloodstream. This is a task blood capillaries can't manage because lipid droplets are too large. Two, they return small plasma proteins that escape capillaries back to circulation, conserving critical molecules like albumin that help maintain oncotic pressure. Three, collect excess interstitial fluid, preventing edema and stabilizing tissue environments for optimal cellular exchange. 4. They deliver foreign particles to lymph nodes, like where immune surveillance occurs. The special flap-like valves, as you can see here in the picture, in the lymphatic capillaries ensure easy entry of fluid and particles, ensuring that the system continually samples tissue environments with pathogens and debris. This is the function bridge between fluid homeostasis and immunity. So as lymph is reclaimed, it is simultaneously screened. Practically, this means lymph nodes are positioned strategically, so near limbs, viscera, and junctions of major drainway pathways, so that microbes and antigens dislodged from any body region are likely to encounter resident lymphocytes and macrophages en route to the veins. So those are the function of lymph. All right, let's talk about lymphatic tissues, lymphatic system in terms of immunity. So the immune arm of the lymphatic system lives in diffuse tissues and encapsulated organs. So MALT, M-A-L-T, which is mucosa associated lymphoid tissue, is unencapsulated lymphatic tissue embedded in mucosa linings of the digestive, respiratory, urinary, and reproductive tracts. So this is where exposure to pathogens is consistent. It's constant. Tonsils and the appendix are examples of rich lymphatic nodules, which are nodules just compact cell clusters. In the ileum, Peyer's patches, let me rephrase. In the ileum, Peyer's patches form group nodules that monitor gut bacteria. Encapsulated lymphatic organs include lymph nodes, the thymus, and the spleen. Nodes are distributed along vessels and filter lymph. The thymus sits in the mediastinum and is essential for T cell maturation during early life. And the spleen, which is the largest of the lymphatic organs, filter blood, not lymph, while also housing immune cells. This distributed layout ensures frontline defense, where microbes first contract the body's mucosa and deeper filtration, where pathogens or debris are likely to pass. Okay, the organization theme here is surveillance, where a particular particle enters through food, air, skin, or blood and will encounter right away lymphoid tissue, primed to respond by engulfing, destroying, or presenting antigens to activate immunity. Okay, let's talk about the actual lymph nodes here. So, lymph nodes are these bean-shaped filters, typically less than 2.5 centimeters long, and they are situated in chains along larger lymphatic vessels. They are common in the neck, elbow, and groin, and within the pelvic, abdominal, and thoracic cavities. But they are not in the central nervous system, so you will not find these in the brain, you will not find these in the spine. As lymph percolates through the node's reticular framework, macrophages engulf pathogens, damaged cells, and debris, while lymphocytes, these are B and T cells, they recognize and respond to antigens. So this makes nodes both filters and immune checkpoints. Structurally, multiple afferent vessels bring lymph in. It slows as it transverses sinuses, allowing time for phagocytosis and antigen presentation. Then lymph exits via fewer efferent vessels, creating basically a useful bottleneck. In addition to filtration, nodes are the site of lymphocyte production, and this goes alongside uh, red bone marrow. They're also made there. And they support clonal expansion during immune activation. So, clinically, enlarged or tender nodes can signal local infection, while firm, non tender nodes may raise concern for malignancy. Okay, so nodes, you know, big time checkpoints for the lymphatic immune system here. Okay, so lymph nodes. All right. So let's talk about the thymus. So the thymus, this is a soft, you see it right there under the trachea. Um, this is a soft bilobed organ, and it's divided into lobules. It is the most active and largest in infancy, and childhood. Then it begins to shrink at puberty, and is actually quite small in adults. So inside, many cells are thymocytes. These are immature lymphocytes, most of which are inactive. A subset matures into functional T lymphocytes, so these are T cells, through a tightly regulated process that selects for cells able to recognize foreign antigens without attacking itself. The thymus here, it produces thymosins, which are hormones that promote T cell maturation. Mature T cells then leave the thymus to populate peripheral tissue and organs where they orchestrate cell-mediated immunity. So this is identifying infected or abnormal host cells. So loss or dysfunction of the thymus in early life leads to immunodeficiency because the adaptive immune system lacks a full repertoire of T cells. Conversely, proper thymic education is centered to self-tolerance, reducing the risk of autoimmunity. So basically, think of the thymus as a training academy. It helps shape the T cell population that the body will rely on for decades in the future. So even after the organ itself has, shrunken down. Okay, the other organ here, the spleen. Now, this is the largest lymphatic organ, and it is in the upper left abdomen. It resembles an oversized node, but it does not filter lymph, it filters blood. So there's two tissues here. There's white pulp, which is rich in lymphocytes and macrophages. And then there's red pulp, which is packed with red blood cells and also macrophages surrounding venous sinuses. Functionally, the spleen filters blood to remove pathogens and debris, recycles worn out erythrocytes, and provides a site where lymphocytes can detect antigens and mount responses. Its robust phagocytic activity helps to keep the bloodstream clean. All right, so because the spleen stores and processes blood components, injury can cause significant bleeding. All right. All right, so here we go. In histology, the contrast between white pulp and red pulp is pretty clear. Conceptually, pair the spleen with nodes. Both are filters with immune staff, but nodes handle lymph, while the spleen controls blood. That is the big difference between um, the nodes and the spleen here. Okay, a little study guide for you guys looking at the organs of the lymphatic system. Okay, so that's the first part of the chapter. Okay, now we're going to look at immunity. So essentially, immunity is just your body's defense against infection. Okay, it's there to defend us against pathogens. Okay, pathogens by definition are disease-causing agents. These can be bacteria, viruses, fungi, spores like fungi. So anything that would come into our body, worms, right, to cause us harm. Two big ones here are viruses and bacteria. Okay, viruses have a simple structure. Okay, that consists of a nucleic acid, could be DNA or RNA, and then they have a protein coat. So what they will do is they will get into our cells, and that will help them produce their own proteins to reproduce. And then bacteria, these are single simple cells, and they also get into our system. They multiply, and they cause some serious infections. Staph and strep are two of the common ones that tend to affect a lot of people. All right, so immunity is the body's ability to keep pathogens out or destroy any that get in. It's twofold. We use two coordinated mechanisms. First is innate or non-specific defense. This is defense that we're born with. So think broad, fast protection. Intact skin is a barrier, stomach acid that kills microbes, fever that slows pathogens and boosts defenses, plus mucus, cilia, inflammation, and phagocytes. And then second, we have adaptive or specific defense. This is targeted protection. Here, lymphocytes recognize particular foreign molecules called antigens. B cells make antibodies, T cells attack infected cells. Most importantly, adaptive immunity remembers, so the next time an encounter happens, it is faster and stronger for the response. Together, the innate system buys time, and the adaptive delivers precise and precision in its immune defense. All right, let's talk first about the innate defenses. We'll start with mechanical barriers. So these are our first line of defense. Simple physical structure that stops germs before they get in. Think of them as the body's no entry signs. So skin here is the big one. It's tough, layered, dry, and it forms armor that separates your internal tissues from the outside world. If the skin stays intact, most microbes never even meet living cells. At openings where we must interact with the environment, we switch tactics. Mucous membranes in the nose and mouth produce sticky mucus. That mucus works like flypaper. Dust, pollen, microbes get trapped instead of reaching delicate tissue. So in the respiratory tract, tiny hair-like cilia beat in coordinated waves. Their job is to move the mucus blanket upward, carrying trapped germs away from the lungs so it can swallow or cough them out. At the front door of the airway, nose hairs provide a coarse filter, attaching larger particles as air flows in. We are also relying on constant flushing. Tears rinse the surface of the eyes. Saliva helps with microbes out of the mouth and off the teeth, and urine flows through the urinary tract, physically sweeping away bacteria before they can stick. Put together, these barriers don't kill everything, but they do prevent, contact, trap, and remove most things. That reduces the number of invaders dramatically and buys time for deeper defenses to act. So those are our mechanical barriers. All right, our next innate defense is inflammation. So inflammation is the body's rapid local response to injury or infection. Okay, when inflammation occurs, we will see 4 signs. We'll see redness, swelling, heat, and pain. The redness comes from vasodilation. So that is our body sending more blood to that area. Swelling happens because leaky capillaries left fluid and immune cells Swelling happens, let's start over, because leaky capillaries let fluid and immune cells move into the tissue. Okay, heat reflects the arrival of warm blood from deeper in the body, and pain occurs as swelling of chemical mediators stimulate nearby nerves. The purpose here is to wall off the problem so it doesn't spread, and to set up defense and repair. So next comes the white blood cells. First you have your neutrophils, then your macrophages. So fibrin will also show up, form a mesh that helps trap microbes. Okay, we talked about fibrin when we looked at the blood vessels forming a mesh to try to clot blood. They also form meshes here in order to try to trap microbes. Once the threat is controlled, phagocytes clean up dead cells, and then finally new cells replace what was lost and the tissue begins to heal. Natural killer cells, NK cells. So unlike B and T cells, they don't need prior exposure or training to recognize a threat. They survey nearby cells for signals of not self or stressed self, and when something looks wrong, like a virus infection or a cancerous cell, they act immediately. Okay, they go right after the problem, and they do not perform phagocytosis, so they do not eat. They release perforins. Now, these are small proteins that are inserted into the target cell membrane, and then they punch holes. Those pores collapse the cell's internal balance, water rushes in, and the cells burst. This is a process called lysis. Natural killer cells can also deliver partner enzymes through those pores to finish the job if need be. So those are your natural killer cells. They do not need to recognize the invader. They just go after it and they punch it through. They punch holes through the invader's cell membrane. All right, the next innate defense, phagocytosis. So phagocytosis literally means cell eating. It's how certain immune cells engulf and digest foreign particles, bacteria, dead cells, and debris, for example, and then they remove them from the body. It occurs within blood vessels and in tissues where blood is filtered, especially the spleen, liver, and bone marrow. The key phagocytes here are neutrophils and monocytes. Neutrophils are the fast first responders. Monocytes leave the bloodstream, mature into macrophages, and become long-lived cleaners in tissues. Damaged or infected tissue releases chemical signals that create a gradient. Phagocytes sense this and move toward the source, a process called chemotaxis. After arrival, they bind to the target, internalize it in a vesicle, fuse it with enzyme-filled lysosomes, and then they destroy it. Together, monocytes and tissue macrophages form the mononuclear phagocytic system, a distributed cleanup crew stationed throughout the body. Their work limits spread, clears the battlefield after inflammation, and prevents antigens to lymphocytes to jumpstart the adaptive immune response for lasting protection. Okay, the innate defense. Next one, fever. We all feel this when we get sick, but it's not random. There's a purpose here. So when a virus or a bacterium triggers the immune system, it activates lymphocytes and other cells that release interleukin 1 or IL-1. IL-1 signals the brain's thermostat to raise the body's set point, so the temperature goes up. But what's the point of raising the heat here? Why even do this? There's three big reasons. First, it slows germs down. Many bacteria and fungi grow poorly at high temperatures. Second, it starves germs. The liver and spleen keep hold of their iron, making less iron available for bacterial multiplication. Third, the fever boosts our defenses. A slightly higher temperature speeds phagocytosis, helping the white blood cells eat and clear pathogens faster. So a fever is strategic. It buys us time, weakens our invaders, and powers up our immune cells. Mild to moderate fevers are often beneficial. Of course, extremely high or prolonged fevers can be dangerous. But in most infections, a modest fever is part of the body's built-in plan to help us win the fight against this pathogen. All right, study sheet for your non-specific, your innate defenses. Okay, so now we move on to adaptive defenses. Okay, so the adaptive specific defenses. So these are the body's targeted protection system. So instead of broad general barriers, this part aims at specific pathogens or their toxins. The idea is specificity and memory. When we're exposed to a microbe the first time, the adaptive system builds a custom response. Then it remembers that invader, so the next encounter is faster and stronger. That's the basis of our immunity. The trigger is an antigen. So an antigen is a molecule on the surface of an invader that the immune system recognizes as foreign. Lymphocytes are B and T cells, and macrophages work together to recognize antigens, activate, and coordinate the attack. So there are two types of adaptive defenses. First is the cellular immune response. That is T cell driven. Cytotoxic T cells identify infected or abnormal body cells displaying the antigen and destroy them directly. Helper T cells release the signals that organize and boost the entire response. Then we have the second type, the humoral immune response. This is B cell driven. B cells become plasma cells that make antibodies. Antibodies bind to antigens like tags, neutralizing toxins and marking invaders So other immune cells can eliminate them. So essentially, T cells attack the problem at the cells. B cells mark the problem in the fluids, and memory makes the next response quicker and stronger. Okay, so this is the line of defense, and we've covered everything up until this third line. Okay, and that's what we're going to talk about here. Okay, study sheet for T and B cells. All right, T cells and cellular immune response. So when a T cell gets activated, it starts with antigen with an antigen presenting cell, also called an APC, such as a macrophage. The APC engulfs the invader. By phagocytosis, it digests it, and then it displays a small antigen fragment on its own surface. Those fragments sit in a special holder protein called MHC, the major histocompatibility complex. A passing T cell constantly scans these displays, and when a T cell specific T receptor matches an antigen MCH complex, the T cell activates. An activated T cell releases cytokines, which are chemical messengers that coordinate the rest of the immune response, telling the other immune cells where to go and what to do. From there, different specialized T cells take the lead. We have helper T cells. They're the signal boosters. They secrete cytokines and activate B cells to produce antibodies, and they stimulate cytotoxic T cells to expand the attack. So then we have cytotoxic T cells. They're the hit squad. They recognize body cells displaying viral or tumoral antigens and kill those cells directly. We have our memory T cells. These are the record keepers. They remain in the body long term, so on the next exposure to the same antigen, the response is faster and stronger. Then we have regulatory T cells, and they're the brakes. So after the threat is cleared, they dial down their response to prevent bystander damage and reduce the risk of autoimmunity. So without these, we very likely would attack ourselves, which is not great. So here's essentially what happens. APC presents on MHC, then the TCR recognizes, T cells activate, cytokines coordinate. Okay. Helper cells, they help out. T cells, they kill. Memory cells learn and prepare for the next time. And regulatory cells stop the attack when it's all over. So B cells and the humoral immune response. B cells are activated when they encounter their specific antigen. That's the starting signal. But to fully activate, B cells must always need help from helper T cells. When a helper T cell recognizes the same antigen, it releases cytokines, the chemicals go signals B cells are waiting for. With those cytokines on board, the B cell proliferates, it clones itself into a large population of identical cells. From that expanding clone, two major paths emerge. Some of the new B cells become memory B cells. Their job is long-term immunity. They remember this antigen, so the next exposure triggers a faster, stronger response. The other route is that the members of the clone become plasma cells. Plasma cells are the antibody factories. They produce and secrete antibodies that are specific for the activating antigen. Okay, those antibodies then circulate in the blood and lymph, bind to the invader, neutralize toxins and viruses, and tag microbes, so phagocytes, and can eliminate them. Because these antibodies travel in body fluids, this pathway is called the humoral immune response. Humoral meaning carried in fluid, like blood and lymph. So here's how it goes. Antigen encounters, the T cell help, the B cells start cloning, memory B cells remember, plasma cells, they move to the antibodies dispatched at the infection site. Okay, here's a visual on how the T and B cells are activated and the B cells clone themselves. Okay, just the next step in this cloning process for the B cells, where they become, either antibodies or they become the memory B cells. And a study guide for you guys. All right, we keep talking about antibodies. Like, what are antibodies? So antibodies are Y-shaped proteins made by our immune system. Think of them as search and tag tools. They bind specific antigens and mark those invaders so immune cells can finish the job. Structurally, each antibody has 4 chains. It has 2 heavy and 2 light. The tips of the Y are the variable regions. They differ in amino acid sequence and fit only their matching antigen. The rest is the constant region, which recruits the right helpers after binding. So we have 5 major classes of antibodies. IgG. Okay, this is 80% of our antibodies, give or take. So this is the workhorse in the blood and tissues. They're great against bacteria, viruses, and toxins. They cross the placenta to protect the fetus, and they activate the helpers. IgA, roughly 13%. This is the mucosal and defender in your tears, saliva, mucus, and breast milk. And this guards the body surfaces and secretion. IgM, about 6%. This is the first antibody produced in a primary response. Excellent at agglutination and complement activation. So here you want to think early infection, detection, and reaction. IgD, okay, less than 1%, sits on B cell surfaces as a receptor, especially important in early life for B cell activation. And then finally, we have IgE. Okay, these bind to mast cells and basophils, and they are essential for allergic reactions and defense against parasites. They trigger histamine when cross-linked by antigens. Okay, so those are your 5 types of antibodies. So essentially, antibodies neutralize, clump, and activate the complements, so the people that are going to come in and kill them, using precision tags that turn recognition into elimination. Okay, so here's the structure of the antibody that we just discussed. Study sheet on your antibodies. Okay, let's talk about the action of these antibodies. So antibodies deal with antigen in 2 main ways. Okay, there's a direct attack and activating complement. First, the direct method. Okay, agglutination is one of the direct attacks, and this is clumping of the antigen bearing particles. So there could be cells or microbes. So they're bigger targets and easier for phagocytes to eat. Second one here is precipitation. And this is similar, but happens with soluble antigens. So antibodies bind and make them insoluble, so they settle out and can be cleared. Essentially, they sink them. And then neutralization is where antibodies cover the toxic parts of the molecule or a virus, blocking them from the ability to bind or harm cells. So they neutralize them. They turn them into the threat into harmless. So those are the three direct attacks by antibodies. So next we have the complement system. This is usually more powerful than direct attack alone. It's triggered especially by the IgG and IgM bond to antigens. So once the complement proteins activate, a cascade produces multiple effects. Glutination, again, clumping of infected cells. Opsonation, this is coating the target to make phagocytases faster and easier. Chemotaxis, we've seen this word before. This is sending chemical signals that attract neutrophils and macrophages to the site. Lysis, we've seen this word before. Building a membrane attack complex that punches holes in antigen membranes, causing them to burst. And then we have neutralization. Again, altering viral structure so viruses can't infect cells. Okay, and the third way here, not the main way, but it is a third way, is inflammation. So this is stimulating local changes that wall off the area and help prevent spread. Okay, so those are the antibody actions. All right, so let's talk about primary and secondary immune response. The primary immune response happens the first time that we meet an antigen. The B and T cells that match it become activated. B cells turn into plasma cells that first release IgM, then IgG. You usually see the first antibodies in about 5 to 10 days, and they persist for several weeks. Importantly, the first response also generates memory B cells, which is our long-term record of the invader. On re-exposure, the secondary immune response kicks in. Because memory B cells and memory T cells are already in place, the system responds much faster and much stronger. High antibody levels appear in 1 to 2 days and can remain for months to years. This is why reinfections are often milder and why boosters work. They refresh and expand those memory pools. The big picture, primary builds the blueprint in memory, secondary uses the memory to mount a quicker, more powerful, long-lasting defense. So these memory B cells can persist for decades. Okay, this is just a chart showing the quickness and the strength of the immune responses. So the days after the exposure to the antigen, we see 15 days at the height of the first exposure, and then 7 days, 7, 8 days on the secondary exposure when it's at its height. Okay, concept of autoimmunity. Autoimmunity is when the immune system attacks the body's own tissues, attacks itself. The core problem is a loss of non-self discrimination. The body produces auto antibodies, antibodies directed against self molecules, and cytotoxic T cells can also target and damage normal cells. So, because different tissues can be targeted, autoimmune disorders vary widely in which organs are affected and in symptoms. As for causes, there are several theories, including molecular mimicry against infections, breakdown of tolerance during lymphocyte development, bystander activation, and regular T cell dysfunction. But there has yet to be a single cause scientifically established. So we really don't know. We just have theories. The takeaway is autoimmunity reflects powerful defenses turned inward, leading to chronic inflammation and tissue injury. Okay, some autoimmune disorders on a study sheet, and that's the end of the chapter. Okay, so thank you guys for listening, and we will, get back to class next Wednesday. Thank you.